forget to wait for the recording. Hi, everyone. Welcome to another week of the Water Resources Research Center Fall Seminar Series. And this week we have Dr. Kasey Barton, who's going to be talking about seedling drought tolerance in a changing climate. So to introduce our speaker, Dr. Kasey Barton grew up in Colorado and received a BS in biology from Stanford University. After college, she was hired as a post-bac technician at Wesleyan University in Connecticut, where she worked on experiments investigating maternal effects in seedling drought tolerance. Dr. Barton returned to Colorado for a PhD in ecology and evolutionary biology, focusing on ontogenetic patterns in plant chemical defenses. She received the NERC postdoctoral fellowship and gained experience and training with research synthesis and meta-analysis at the University of London. In 2010, she joined the botany faculty at UH Manoa and has loved exploring the functional ecology of Hawaii's endemic plants and hopefully contributing towards their conservation and restoration. With that, I'll let you take it away.
Okay, well, thanks everybody for coming out to hear about seedling drought tolerance. This is something we've been working on in my lab for about 10 years now. So I'm going to do some sort of highlights of some of our most exciting results. All right, so plant-water interactions are obviously really important, and plants are really driving some of the major fluxes of water in the environment, including different pathways of water uptake, and then also, of course, the return of water to the atmosphere through transpiration. So if we're thinking about these ecosystem scale water dynamics, then it obviously makes a lot of sense to focus on the large trees like this ohia lehua that you see in the image, because these large trees are the ones that are really driving these dynamics at the ecosystem scale. On the other hand, if we're looking at this from the perspective of the plants, then we have to consider not only those adult trees, but really all the life stages, because they will also contribute to the population dynamics and help us understand the water effects that might be having on plant population dynamics. So in particular, from the plant perspective, it's really critical that we consider seedlings. And that's because seedlings are much more sensitive than adult plants to water limitation. And this is really for two primary reasons. The first is that seedlings are small. And because they're small, they have limited root biomass and therefore relatively shallow root depth. So they're only able to access water really in the surface soils. And also because they're small, they tend to have relatively little stored reserves, particularly non-structural carbohydrates. And these play important roles in osmoregulation in order to respond to in a plastic way to maintain water uptake. And they can also provide carbon during periods of low gas exchange. So being small makes plants inherently sensitive to water limitation. But there's another evolutionary reason that seedlings tend to be more vulnerable to water stress, and that's because they've been selected to have an acquisitive growth strategy. So what this means is that there's been selection for a strategy that includes the production of short-lived tissues that are usually associated with high rates of photosynthesis, but which don't last very long and are also quite vulnerable to water damage, water stress damage. And that's because of reduced leaf venation, minimal water storage, having a low leaf mass per area. And the reason that there's been selection for this acquisitive strategy is really twofold. One, because the easiest way to escape the vulnerabilities associated with small size is to grow. So there's been selection for fast growth at the seedling stage so that outgrow this vulnerability of being small. And the other reason comes from competition. So small size advantages during the seedling establishment phase lead to imbalances in competition, and that can totally determine whether or not a seedling establishes. So because of this, across most species, there's been selection for a really acquisitive growth strategy, even though that corresponds to a relatively risky hydraulic strategy. And that makes seedlings particularly vulnerable to water stress. And this is important because usually when we think about species distributions and the kinds of climates that plants encounter or occupy, we base these on adult plant distribution and presence. And actually, the seedling climate niche is usually much more narrow than that of the adult stage within the same species. And so just by looking at the species distribution of adult plants, we can't infer the climate thresholds of the seedling stage. And that makes it really difficult to predict how climate change is going to affect seedling recruitment, which is fundamental to the population stability of plants. So this means that we have to explicitly look at and try to characterize the climate niche and the aridity thresholds at the seedling stage. And this is clearly important. There are lots of observations and reports about how many different plant species are encountering reduced seedling recruitment and relatively few small and young plants out in natural communities. And this is indicating that climate change is probably moving them beyond what is within the recruitment climate niche. So these are early indicators of climate change stress, which we may not even observe if we were just focusing on adult plants, because the adult plants can tolerate these changing climates more than the seedlings usually can. And in addition, this can be particularly complex to investigate because the aspects of climate, and particularly with respect to water, may be slightly different in how they affect seedlings and adults. So for seedlings, we may be looking at climate factors like the frequency of precipitation, the number of consecutive days without rainfall, the extent of extreme droughts. Sometimes that's paired with extreme rain and floods, which is also really stressful to seedlings. And of course, decreasing humidity, because that affects vapor pressure deficits. So these are the climate factors we really need to be paying attention to try to understand how seedlings are going to be responding, and then therefore whether recruitment is being affected by changing climates. So this is the work that we've been doing in my lab. We've been focusing on the seedling stage and trying to understand how Hawaiian plants are responding to increasing drought. And we're particularly interested in variability. So we're really trying to capture and characterize how variable seedling drought tolerance is across multiple scales. So at one scale, we're interested in species variation. So how species differ in their capacity to tolerate drought at the seedling stage. We're also interested in patterns within species, especially widespread species that grow across climate gradients. So this is thinking about local adaptation to localized climate across gradients. And also looking at how drought tolerance varies within an individual plant. So that incorporates developmental change, which captures the seedlings and how they may differ from older plants, but also includes more immediate responses to how plants acclimate to decreasing water availability through phenotypic plasticity. And then finally, a lot of this work incorporates functional traits and trait analyses, because this allows us to get mechanistic insights into what could be driving all of this variability. And that helps us make better predictions about perhaps which species or populations are likely to be more drought tolerant in the future, and also just helps us understand how these plants are working in response to water availability. So we've been focusing on lots of plants, specifically trying to capture a lot of diversity, as much diversity really as we can get access to. So this includes different kinds of growth forms, different kinds of plant families. And this is just a selection of some of the native species that we've been investigating seedling drought tolerance in. We've also included a number of invasive species in these trials in order to investigate questions about whether climate change may potentially lead to increased recruitment of invasive species if, for example, they're more drought tolerant than native species are, or vice versa, if climate change may actually be a mitigating factor, reducing invasive plant distributions. And we've been doing this in both the field and the greenhouse. So I think there's really good benefits to both approaches, and I actually really like combining field and greenhouse experiments together because they can allow us to investigate different aspects of seedling drought tolerance. So on the one hand, we often prefer to do field experiments because that includes all of the real world complexity that the plants are actually experiencing out in natural areas. And that's really important because water availability and water stress to plants is totally context dependent. And so all of these localized factors like soil substrates, temperature, neighboring species identity and density, whether there are herbivores, et cetera, all of those other ecological factors can influence the extent of water limitation to plants, and therefore also how the seedlings are responding to that. So that's a really important aspect of field experiments, but it also makes it really hard to know when we're measuring seedling responses to drought, how much of their response is actually driven by water limitation versus all of these other ecological factors. So by combining field with greenhouse experiments, we can use more controlled conditions in the greenhouse to carefully calibrate water availability and therefore more precisely measure how the plant's responding to that in particular. And that allows us to compare across species more easily because we've held everything else constant. And it also allows us to do really careful functional trait analyses, which are more challenging to do under field conditions. So there are advantages and disadvantages to both of these, which is why really I think the ideal includes both. Because, like I said, plant drought tolerance and the extent of water limitation is so context dependent, one of the biggest challenges is knowing to what extent we can generalize the findings of an experiment. So we often think that field experiments are more informative because we can generalize more easily. But I don't think that's actually the case. It's always going to be true that wherever we measured seedling drought tolerance in the field, it really only applies under those particular conditions, that particular site and its soil substrate and its elevation and the neighboring plants and all these other things. So extrapolating from any experimental condition in the greenhouse or the field is going to be a major challenge, especially if we don't totally understand how much those other factors may moderate the effects of water limitation. So I think we always have to be really careful about this and mindful that when we're measuring seedling drought tolerance under, say, controlled conditions, which we do in the greenhouse, that's a measure of potential tolerance. So it's the intrinsic capacity of a species to maintain fitness under drought. And in this sense, fitness is the biological definition. So really, I mean survival and growth for seedlings. But how well potential tolerance actually informs us about realized tolerance is always difficult to know. Realized tolerance is the actual seedling maintenance of fitness under all that real world complexity in the field. And the real problem is that realized tolerance is going to be variable across all these different environmental factors. So that I think is really our biggest challenge, trying to understand how all these different factors contribute to the capacity of plants to tolerate drought and what that means out in the field in real populations for recruitment under climate change. And that's the challenge that we're trying to get to. That's what we're working at. And this is, I'd like to say just right up front, a huge group effort. So we've been doing this for a long time, tons and tons of trials, many thousands of plants that we've grown. And this has been a huge effort by so many wonderful students that I want to acknowledge. These are undergraduate students, graduate students, postdocs, and even my son, you can see he's been working on this since he was in kindergarten. He's now in high school. So this has really been a big team effort. And what a fun job to get to work with all these wonderful students. So I'm just going to highlight 4 of our key results, trying to keep this at sort of like showcasing some of our most exciting results. The first is just that there's tremendous variability across species in their seedling drought tolerance. So I'm going to focus on a recent study where we compared 32 different taxa. These included taxa that were sampled across 3 different islands. This is hard to see, but I'm showing you those sampling sites in the map and the phylogeny there as well. This includes 8 endemic species, 11 non-native species, 5 indigenous species. And for several of these, we also have multiple populations that we sampled within or across islands, trying to get at this within species variability. We purposely tried to capture a broad phylogenetic diversity. So we have 15 plant families, and our initial phylogenetic analyses indicated a weak or negligible signal for phylogenetic effects on seedling function, seedling drought tolerance. So we can just exclude phylogeny from the analyses that I'm going to show you. So what we did, this was in the greenhouse, and we simulated drought on seedlings that we grew from seed. So this is an Ohia lehua seedling, and this is a little bit bigger. So this one's in drought. We usually initiate drought at the 5 to 6 leaf stage, and we make that consistent across all the species. So it takes some longer or less time to get to the 5 to 6 leaf stage, but that's when we start drought. We initiated a pulse drought, which means we withheld any water from the drought treatment plants for 2 to 3 weeks. We ended the drought when 50% of the drought treatment group were severely wilted or had died. And then we had a control group that we watered every day for this period. After that pulse drought, we watered all the plants again for a recovery period of 4 weeks. And then we harvest both shoots and roots so we can get a total biomass estimate of fitness. And then what I'm going to show you are analyses that try to synthesize across species. So within each species, I'm capturing that difference between drought and control seedling growth with an effect size called the log response ratio. And so this is just a way of calculating the difference that incorporates the variance within each of those species, allowing me to then synthesize across species using meta-analysis. So here are those results. And what I've done with the log response ratio effect size is I've back transformed that into a percent effect of drought on growth. So these are percentages on the y-axis. So 0 in the middle means there's no difference in the total biomass between control and drought plants. A percent that's positive is a percent increase under drought. So those are plants that actually grow better under drought compared to control conditions. And we actually have 3 native species that actually did better under drought. So these are really drought tolerant plants. In fact, they perform better when there's less water. But what's more typical is these negative percent effects of drought. So these are decreases in growth under drought compared to control. Each of these dots or symbols is a species. It's hard to see, but the diamonds, which are green, but you may not be able to see that. That's why they're diamonds, are the invasive species. And then the blue circles are the native species. I'm not differentiating endemic and indigenous because we didn't detect any difference between them. So what you can see is that there's this huge range from these 3 species that actually do better under drought to quite a few species that are reduced by about 30% in growth, all these ones in the middle here. And then we see down here the species that do very poorly under drought, up to about a 60% decrease in growth. So that's interesting. There's a ton of variability. And we see the native and invasive species sort of all across this range. I'm just highlighting a few things. The 2 most drought tolerant invasive species are guava and Formosan koa. And then down here, I've highlighted these 4 species that are the least drought tolerant, which is actually 3 invasive species and just 1 native species. So quite a lot of range, right? If we group these now by their origin, so native versus invasive, we see that actually on average, the native species can tolerate seedling drought better than the invasive species. Although this is only marginally statistically significant, right? P-value of 0.0789. But nonetheless, this is perhaps good news that at least focusing on this assemblage of species, that there's no indication that climate change is potentially going to tip things in favor of the invasive seedling establishments. We suspected that we could relate the seedling drought tolerance to the climates from which we sampled these species. So we looked back at where we collected the seeds, the source populations for these seeds that we grew up in the greenhouse, and we then got the climate data for those sites and tried to investigate relationships between those source climates and seedling drought tolerance from our experimental trials. And we actually don't detect any significant relationships. So mean annual rainfall, mean annual evapotranspiration, and the aridity index, none of them predicted seedling drought tolerance across these species. So that could indicate that there's other things leading to variation among these species. And it could also mean that we're not capturing the right climate variables. So you notice none of these are those ones that I identified, number of days without rainfall, say, or some of the factors that we think are really important for seedling evolution. So there's probably more work to do here, but this initial glance doesn't have us understanding how climate at the places where these populations have evolved is explaining their variability in seedling drought tolerance. One other possibility is that it may be easier to detect this relationship with climate if we look at variability within species as opposed to across species. So that's the next example that I want to share with you. So in this case, we're going to be looking at the role of local adaptation via seedling drought tolerance and look at how that may be affected by current and ongoing climate change. So for this, we're looking at Ohia lehua. So I'm sure you all know Hawaii's most widespread and abundant tree species. I'm showing you here its species distribution model. Obviously, this is a climate generalist, right? So almost every climate is suitable for Ohia lehua. And that might lead us to be pretty confident that with climate change, the species is going to be resilient because any new climate that it encounters is still within its climate envelope. On the other hand, if we zoom in and consider populations and the localized climate, it's probably not the case that every population has the same broad climate threshold or tolerance that the species does. And because adaptation happens at the population scale, that's what we wanted to investigate, how local adaptation at the population scale may be affected by climate change that's happening also at that local scale. And of course, because Ohia is a foundation species, its resilience and stability under climate change is super important, right? There's so many plants and animals that depend on Ohia for critical habitat. And so this is a really high priority to understand how it may or may not be affected in terms of seedling recruitment under climate change. So to investigate this, the best way to test for local adaptation is with reciprocal transplant experiments. So that's what we did. And here on the island of Oahu, and we captured a pretty good precipitation gradient, so mean annual rainfall gradient of 3,500 millimeters. And across that gradient, we identified 27 populations. I'm calling these populations of Ohia. We didn't actually detect or investigate if they are genetically differentiated, but we have 27 sites across this gradient where Ohia are in the canopy. We collected seeds from those trees. In the lab, we made sure that the ones that we were putting back out in the field were filled so that they had embryos, they were viable seeds. And then we counted them out into 50 seed little groups. And we did a full factorial reciprocal transplant, which means seeds from all 27 source populations went back out into all 27 sites. And we did 3 replicates of those 50 seeds in each of those source by site combinations. So in total, this was 109,350 seeds that we sowed back out into the field. And then we sowed these onto cushion moss, which is what I'm showing you here, because that is where Ohia often germinate into. So it's a pretty moist microenvironment, which facilitates seedling establishment for Ohia, because these are dust seeds, which you can see next to that quarter. They're super, super tiny. They're really vulnerable. So then we went back out to our sites every 2 to 3 weeks, and we counted the number of seedlings that were germinating in those plots. And the way to test for local adaptation is to look for a home site advantage. So if a population is locally adapted, then it should have higher performance in its home site than it has at any away sites. So that's pretty easy to test. And we did that both in terms of the percent germination, so the total number of seedlings that germinated in these plots, and also the percent establishment, which were the number of seedlings that survived to the end of the experiment. And in neither case did we detect greater performance at the home site compared to away. So comparing for germination or establishment. So this had us thinking that maybe they weren't locally adapted. But on the other hand, we looked at the rainfall patterns for the year that we did the experiment. They didn't correspond in a very obvious way with the historical patterns of rainfall. So we had that 3,500 millimeter rainfall gradient, but the rainfall during the year that we did the experiment did not have the same kind of ranking. So we thought, oh no, there's something going on. There's a decoupling of historical and contemporary rainfall patterns. So we wanted to see if that could be driving some of this variability. So to get at that, we developed a conceptual model. So our idea was that, that we expected that actually another problem could be that all populations may have better seedling performance when there's more water available, because seedlings are just inherently water limited, like I discussed at the beginning. So we suspected that across every population, all 27 populations, that they would all do the best in the wettest sites, because those are the ones that are best for seedlings. So what we are showing here, each of these lines is meant to be a population response in terms of seedling germination or establishment with respect to the rainfall gradient in the year of the experiment. So on the x-axis is the 2015 precipitation. That's the rainfall during the experiment. So some of our sites were dry and some were more wet, right? That's the gradient during the experiment. We thought every single population would do increasingly better when there was more rain, and we'd see a positive response. However, we suspected that populations from historically dry sites, which I'm showing with the hatched line at the top here, would do best in wet sites, but would also do relatively well in dry sites, because presumably they've adapted to those dry conditions because they came from a historically dry site. So we suspected that those populations from dry sites would have a shallow slope, and that the steepest slopes in response would be from historically very wet sites, because they would do the worst in dry sites because they're historically adapted to wet conditions. So I'm showing you just 2 lines here. Of course, in reality, we have 27 lines, right? Because we have 27 populations. But this is our theoretical model. We then suspected that we could calculate the slopes of these responses and look at the relationship between those slopes, which I'm showing now in the y-axis, and again, with respect to the historical rainfall at the source population. So remember, I thought the steepest slopes would be from historically wet sites. So here's historical, these are the wettest sites, they have the steepest slope. And that we get increasingly more shallow slopes as we went to increasingly drier sites, because they would do quite well in wet or dry sites. Okay, so this one is showing us the patterns within each source population across sites. And this one is looking at the pattern across the populations. Okay, here are the real data. This is what we actually found. So here are all those 27 lines. You can see they are variable. Some of them are showing this predicted increase, but actually a surprising number are showing a decrease, suggesting that actually for these ohia, they did better in dry sites than wet sites. That is a puzzler, and it could mean that there's other threats that are correlated with wet conditions, like seedling herbivores or pathogens, things like that, which are often more common in wet conditions. Nonetheless, if we calculate the slopes of these 27 population responses, here we do get the significant positive relationship that we predicted. So those positive lines that we're seeing over here are indeed from our wettest sites. Those are the ones showing a really big decrease in seedling performance under dry conditions, and that this gets increasingly more shallow, or actually increasingly more negative as we get to drier conditions. So this is consistent with local adaptation, but the problem is that it's consistent with local adaptation to historical conditions. It's not leading to a home site advantage because the home site conditions no longer resemble the historical conditions, because climate change is already leading to a decoupling of local adaptation. So that's what we conclude to be the result here, that there is evidence for local adaptation, but that doesn't lead to home site advantage. And this could be why when we collect seeds locally and put them out, they may not be doing very well, because the climate no longer resembles the climate that these populations have adapted to in the past. As always, we need to consider in the field that there could be other things going on, right? Like the seedling and pathogen, seedling herbivore and pathogens that I mentioned. So we thought, to confirm really that it's about water availability, we paired this field experiment with a greenhouse trial where we could simulate drought and remove any of those other factors that we think may be contributing to seedling germination and establishment. So for this experiment, we simulated drought, this time in a different way, using a press drought. So a press drought is when the plants get water regularly, in our case, every other day, but we give them less water to maintain them at a set target soil water capacity. So in our case, we reduce soil water capacity by about 20% compared to watering to dripping or field capacity. And that we maintain for a 6-week period. And then at the end, and you can see that here. So here's before the drought, the control and the drought have similar soil water content. This is soil water content. Day 28, we started the press drought. You can see those drought treatment groups immediately drop, and then we maintain them at this level with a little bit of water every other day. These plants continue to be watered every day to dripping. And then after 6 weeks of press drought, we did a terminal drought, which means we remove water from both control and drought groups, and we just see how long they can last until they die. And the reason for this is because we want to know what the threshold is in terms of number of days the seedlings can live before they die. And we can also measure the soil water content, and that gives us an indication of what's the lethal soil water content that then the plants cannot tolerate below that. And by comparing the plants that initially got a press drought versus control, we can see if having this press drought sort of acclimated the plants to drought, enabling them to live a little bit longer when they undergo the terminal drought. Okay, and then we did this across all those 27 populations. So this was a huge experiment that included control and drought treatment groups for seeds from those same 27 populations. And here are those population level responses. So each of these dots is a population mean of those 27, and then the error bars. And what I'm showing you here, always on the x-axis, is the historical mean annual rainfall. That's the rainfall gradient that we collected those seeds from out in the field. And then here are some measures of drought tolerance. So what we can see, this is probability of mortality. So the populations from historically wetter sites had higher, significantly higher mortality under drought than populations from dry conditions, consistent with local adaptation to seedling drought tolerance. And they also, we detected variability in the longevity that they could live in that terminal drought. So again, the populations from wet conditions survived significantly fewer days under terminal drought than the populations from dry conditions. And when they died, the soil was significantly more wet. All of this corroborating the field study, but actually now solidifying the role of water availability in those population level variability and consistent with local adaptation to seedling drought tolerance. So this is really interesting, but doesn't matter. Like on the one hand, maybe not, because if we're getting declining seedling recruitment for a population because it's getting increasingly drier, and the seeds that are coming from those local trees are not adapted to that increasingly drier conditions, ohia lehua might be okay because their winds disperse. These are tiny seeds, so they might blow in from populations that are drought adapted. And so at the species scale, we might predict that it might still be okay because at the species scale, they harbor all this genetic variation in seedling drought tolerance. But if that were to happen, then we'd essentially be replacing those wet adapted populations with dry adapted genotypes. And we'd end up with a more genetically homogeneous species across its whole distribution. And that would be a real shame because ohia lehua is polymorpha after all. And it harbors all of this incredible intraspecific diversity, not just in seedling drought tolerance, but lots of other traits that have biological and cultural significance. So I would argue that the population is the unit of conservation concern, and we should be mindful that we could be losing populations due to reduced seedling recruitment under increasingly dry conditions, and not just feel okay that they might be replaced by drought adapted populations from elsewhere. All right, it's not surprising to detect intraspecific variation in seedling drought tolerance for a really widespread species like ohia lehua. But actually, what is kind of surprising is we've also detected within species variation in seedling drought tolerance in critically endangered species like Shadea obovata. So in this study, we also detected significant variation, and it indicated that the population that's historically from the wettest part of its distribution is significantly more vulnerable to seedling drought than the populations from historically dry places. So this corroborates the idea that this intraspecific variation in seedling drought tolerance for almost any species that occurs across diverse climates is likely to be happening and could be contributing to population stability or the lack of population stability under climate change. Okay, so what about sensitivity to the timing of rainfall? So those are like broad scale patterns of variation. My last two examples are a little bit more mechanistic. So in this case, we're working on ohia lehua again because we focused on that quite a bit for this work. And what we wanted to investigate was whether the frequency or the timing of water contributed to the performance of the seedlings. And so in particular, we wanted to know that if we maintained, or if the seedlings got the same amount of water in total, but they got that in either little bits frequently or big rainfall events infrequently, how that would affect seedling performance. So we tried to design some treatments that would reflect that or simulate that. And I'm showing you those here. So our control group is just watered to 100% field capacity every day. Then we have 3 groups that are in a press drought where we maintain them at 80% field capacity. And they get watered either every other day, once a week, or every other week. So the ones that get watered every other week are getting big amounts of water because they're getting to that 80% FC, but they're only getting it every other week. Whereas these plants in the every other day get just a little bit of water every other day. So the same soil water potential, but different frequency of water. And then we have a pulse drought where there's no water for 4 weeks. And we found that this did significantly affect the plant performance. So this is just total biomass. And what we can see is that, of course, under those regular control conditions, the plants grew the best. The plants that had no water for 4 weeks grew the worst. They were really tiny and actually had really high mortality. And then among those 3 groups where they were maintained at similar soil water potential, but at different frequencies, we do detect significant variation that's really hard to understand. It's not clear why getting it every once a week is worse than getting it every other week. And there was really high mortality too, so it could just be driven by having small sample sizes here. But we paired this with a lot of trait analysis to try to get a better understanding of how the seedlings are actually responding to water limitation, because that can help us then understand what's going on with these different kinds of water availabilities. So this is a model that describes and is informed by a lot of trait analysis and statistical models behind the scenes, but this is just now trying to summarize it for you. And basically what happens for ohia lehua under water limitation is that the first response is reduced stomatal conductance. So they close their stomata, which is a pretty common and conservative response. It's characteristic of the isohydric drought tolerance strategy where the plants try to maintain the water they have to limit water loss through transpiration. The result, of course, of reduced stomatal conductance is that there's also less CO2 uptake, so photosynthesis gets reduced. And that is how we end up with smaller plants that grow less. Here's where something interesting happens. There is plasticity in biomass allocation. So even though total photosynthesis and growth decline, the root biomass is prioritized. So we see a shift to prioritize growth below ground, which makes a lot of sense because roots are the organ that take up water for these seedlings. So that enhances or at least maximizes water uptake under dry conditions. But a consequence of that is that there's also relatively high leaf nitrogen under drought because of that increased root growth. And that leads to a maintenance of chlorophyll in the leaves. And having high chlorophyll under drought conditions increases water use efficiency. And that is statistically linked to survival. So it's pretty interesting. We actually see tons of mortality. So I would say ohia lehua is not very drought tolerant. But the ones that do manage to survive have this really interesting and complex physiological cascade of events and responses that enhance their survival. And that can help us understand the variability that we've been observing in the field across those populations and also with respect to precipitation frequency. And then my last example is trying again to put this into some real world complexity by considering how other ecological factors and interactions could influence or interact with drought stress to seedlings. And for this one, I'm going to focus on herbivory. So we've done some experiments with both mollusk herbivores. So seedling herbivory is another major threat to native plants because there are a number of invasive herbivores that target seedlings in particular. And this includes invasive mollusks like this giant African snail. And also there's lots of mammals that browse seedlings. And so we've done experiments where we actually include the snails to see what they'll eat. And also we've simulated mammalian browsing through clipping treatments of the seedlings. It's hard to tell, but this koa seedling has been clipped on the shoot here. And we've combined these herbivory treatments with drought to try to test whether herbivory influences the capacity of these plants to tolerate drought at the seedling stage. And the species I'm going to share with you for this are shown here. So again, trying to capture a diversity of plant species. And we've used both the press and pulse drought for these experiments. So the press drought was maintained at the 70% field capacity, so they got water every day, but to maintain them at a reduced water availability level. Pulse drought got no water for 4 weeks. And then we simulated herbivory in this particular case with those 50% shoot removal treatments to simulate browsing. And then we have, of course, control plants that were watered every day. So the plants got either only a herbivory stress, only a drought stress, or the combined stress of herbivory plus drought. And that could be herbivory plus the pulse drought and also herbivory plus the 70 FC drought. And then we harvested them shortly after the herbivory treatment 2 weeks later, for which is what I'm calling the early harvest. And then we gave them a bit longer time to grow up after that, which is the 5-week harvest. And I've synthesized these responses across all the species into, again, a log response ratio. And then I've back transformed that to percent effects. So percent lower biomass in stress than control plants. These are all negative numbers, which means there are no examples of species that actually grew more under stress than control. So what's interesting is the plants that encountered only one stress, so just one of the 2 droughts or herbivory, were suppressed significantly less than the plants that encountered both stressors, right? So the 2 stressors has a greater negative effect on seedling growth than one stress alone. And that was detectable at the early harvest, and it was also detectable at the late harvest. But what's interesting about the late harvest is these error bars are much bigger. And that's because we detect a lot of variability among the species and whether they could catch up in growth when given that extra 3 weeks of time to grow. Some of them got increasingly closer in size to the control plants, and some of them got increasingly smaller. So they were really penalized by those early stress events. And we know from, I'm not showing you here, but we tried to disentangle what was happening below and above ground. And the conflict here is that with herbivory alone, plants will allocate relatively more growth above ground to replace that lost shoot tissue. So we see a reduction in root growth under herbivory and a maximizing of shoot growth. We see the opposite, of course, with drought, right? Less shoot growth, more root growth. So when they have both drought and herbivory simultaneously, now there's this conflict between should they prioritize root growth or shoot growth, and we actually end up seeing almost no shift in biomass allocation, which must mean that they kind of cancel each other out. And that makes the plants particularly vulnerable to the simultaneous stress of herbivory and drought in particular. So I think this kind of interaction between multiple stressors is very common. So we're showing it here for herbivory and drought, but of course, competition is likely to have these sorts of interactions, disease, if we consider other life stages, pollination limitation, etc. So the more we can do these simultaneous stress experiments to begin to understand these interactions, I think the more precise and confident we'll be with making predictions for how climate change is going to affect seedling recruitment. So I hope I've convinced you that focusing on seedlings is relevant and important because, after all, to get to these really big trees that are dominating the plant water fluxes at the ecosystem scale, everything starts like this, super tiny, and they have to be able to survive and grow in order to replace these big trees when they die. So population dynamics is totally dependent on seedling recruitment, and because seedling recruitment has such a more narrow climate niche, it's really at risk with climate change. And then all these other global threats are sort of confounding that and making it really hard to predict what kinds of conditions are thresholds or not for maintaining recruitment under climate change. And there's loads more we're doing on this, or we want to do on this. We're sort of just getting started, even though we've already been working on it a while. Right now, we're really interested in field experiments that are exploring climate matching for restoration. So this idea of, with ohia, where we're finding that the plants are not doing best at their local sites, suggests that maybe they'd be doing better at sites that match their historical climate. And that's called climate matching or assisted gene flow. And that's a pretty commonly used strategy for restoration in some parts of the world. So we're interested in exploring that. We're interested in interactions between drought and competition, so kind of expanding from that drought by herbivory interactions. I think other extreme events like flooding, which often co-occur with drought sort of over time, are really important for seedlings. And even what doesn't seem like a big flooding event for a seedling could be a flood. So I think that's really important to add on to this experimental approach. Climate interactions, the ground temperature can be really high, and that can be important to consider, especially when drought and that heat increases are simultaneous. Drought and fire are interacting. One of the things we're trying to do with the functional trait is by identifying traits that predict seedling drought tolerance, we could use those traits as potential indicators of species that are likely to be very drought tolerant, or the converse, to be very vulnerable to drought. So we can then go out and do more species screening through trait analysis to try to identify these vulnerable and tolerant species to seedling drought without having to do such cumbersome experiments. At least that's one of our goals. And as always, I'm happy to take ideas and insights for folks who know lots of other plants and know the plants better than me and different sites better than I do. And it's always great to get input and ideas for other studies that we can do. So thank you so much for your time, and I hope I can answer some questions.
All right, we have a pretty small live audience. So any questions though?
I'm doing a lot of press. Well, I do actually have a question. So that was a very interesting talk. I'm not working, I'm working in environmental engineering, so I don't work with plants, but it was a very interesting talk. So you talked about like potential selection of tolerant species and also like decrease of diversities within a species by drought, and also some selections of general traits. So I was wondering, just curious, like in general, what are there like potential feedbacks of these traits or these selected species to the local soil condition or local ecosystems that may make the this effect more serious, or vice versa?
Right, yeah. That's a great question and complex, right? So I suspect the seedlings aren't themselves contributing very much to sort of the ecosystem, nutrient availability or chemistry of the soil, but the adult trees definitely are through their foliage and through exudates in the roots and from the rain flow through the canopy, etc. So there's a potential mismatch between the identity of the canopy and the seedling community that's below. And so I think for that question, we really need to be looking at what's in the canopy and how they are perhaps altering or affecting the conditions of the soils where the seedlings have to regenerate. And there's definitely some invasive species that we know for sure have allelopathic type effects and also shading effects that are negatively affecting native plant recruitment. And that's one of these community ecosystem scale complexities that's really important, but we haven't been investigating that yet. Totally valid that we could. Yeah. Yeah. Thank you for showing up.
Okay, we have quite a few questions in the chat, so I'm gonna get started reading those. So the first one is, do you think contemporary rainfall data will provide better describing the variance of the seedling source, germination, or home advantage?
So yes, we don't detect a home site advantage, and that's because we think they are locally adapted, but the home conditions don't reflect the historical conditions that they adapted to. So here's the trick: is we have to relate the contemporary rainfall to the historical rainfall, and we have to find the relationships between those. And that's kind of what climate matching is trying to do, is it's trying to identify what was the historical climate, where does that climate exist today, and there's where the local adaptation should go, right? If they're locally adapted to climate. What we're seeing is a decoupling of climate-based local adaptation because climate change is already happening. That's why we're not seeing a home site advantage. I hope that answers the question.
Follow up from that, so asking about other factors to represent or affect the drought, could evapotranspiration be a secondary variable, i.e. a site with high rainfall but also high evapotranspiration, likely not as wet as sites with high rainfall and low ET, or perhaps soil types. Water holding capacity is likely different by soil type.
Yeah, thank you for that. And also, my apologies for oversimplifying it. In that Ohia field experiment where we did this kind of analysis looking for local adaptation, we did investigate mean annual rainfall and mean annual potential evapotranspiration and found the same patterns. So actually, they both corresponded significantly with our population variation. So we concluded that it's sort of, it's really water availability, but we're detecting that through mean annual rainfall and mean annual potential evapotranspiration. Without a doubt, the vapor pressure deficit is important. Yeah.
Next chat question. I was wondering how you think increasing CO2 concentrations, which could increase water use efficiency in plants, could interact with droughts to affect seedlings.
Sorry, could you say that again?
How do you think increasing CO2 concentrations, which would increase water use efficiency in plants, would interact with droughts?
Yeah, so that's a good one. And there's loads of experiments on that, and also on seedlings in chambers. So I think we have some pretty good ideas that in the short term, CO2 enrichment can offset drought stress and enhance performance, but that it's usually a short-lived effect. So it doesn't seem to have like a persistent or long-lasting effect on the seedling recruitment. Although in the chamber, I'm not sure we can call it recruitment, but seedling performance. So I think in the long run, we might be overconfident if we predict that CO2 enrichment is going to offset this dramatically enough to really affect recruitment. I think the water limitation for seedlings is so sensitive, and they can really respond to what seem like almost no effects or decreases in water availability because they're so constrained and they have such a risky hydraulic strategy. At least many forest species, that's true. If we're talking about desert plants, it's probably a different story. So I think the CO2 enrichment is physiologically interesting, but ecologically maybe not going to be so important.
Next one, can you please expand on the linkage between increased chlorophyll in leaves and plant survival?
Yeah, that's come up several times. So I feel like that's a really robust result. We also detect that as a significant predictor across species for salinity stress tolerance, which we've done in the coastal beach dune plants, which is my picture that I'm showing you right now, which is very similar to drought stress in many ways, because salinity stress is mainly osmotic stress. And chlorophyll content is significantly related to drought and salinity stress tolerance across species. And I'm not totally sure, but that is correlated with water use efficiency. So we're interpreting this as having the high chlorophyll content is somehow enabling the plant to overcome that constraints of reduced stomatal conductance and draw down CO2 maybe through, I don't know, epidermal diffusion, and therefore do enough CO2 fixation in order to survive. So this is really linked not just to survival, but also growth under drought. So if we just look at the sizes of the plants under drought conditions, the ones that grow the best, that's predicted by chlorophyll content and water use efficiency, despite reductions in stomatal conductance and net photosynthesis. Yeah, thanks.
Okay. Next commenter says, so much detail and work. My mind is blown thinking about all the cross applications to marine plants and salinity tolerance and groundwater inputs into the marine system. So inspirational for future work. Well, this is just yes, this recording will be available. Okay, next question is, I wonder whether or not there is a difference in moisture dynamics in soil versus moss, and whether precip is accurately representing the moisture conditions of those Ohia seedlings.
Yeah, so that's a great point. And we don't know because we didn't measure in the field the soil water content. We only did that in the greenhouse. And the moss is, in the picture I showed you, like way more beautiful than in reality. Can you see this picture here? This is what our little babies look like. So you do see there's moss here, but there's also a lot of bare soil. So those ohia are germinating. We did sow the seeds on moss, and that was partly to try to keep the seeds in the plot so they didn't just blow away. But these seedlings are definitely rooting in the soil. So I do think that it's not just the moss that's at least informing us about seedling establishment. But I also think that actually our rainfall data in the year of experiment is pretty coarse because we don't have local sensors. This is based on rainfall gauges. And sometimes we had to use the same gauge for multiple sites and this sort of thing, which is why we thought, it's hard to know exactly how robust those field studies are for concluding local adaptation to rainfall availability or soil moisture content, which is why we did the greenhouse experiment, which is the second part. And that really corroborated it in a way that gave us more confidence about the field. But nonetheless, there's a lot of variability in the field. We're still detecting these pretty strong patterns, but within that, there's a lot of variability that's unexplained, which is certainly could be due to mosses playing a role, pathogens, herbivores, shading, all these other complexities. Yeah.
A follow-up question / comment from the previous asker. Also, sorry, I'm not able to identify the people who are asking these questions. I was told that for privacy reasons, I can't give identifying information. But anyway, follow-up is, I'm surprised that Christmas berry has so little drought tolerance given that it occurs in the really low elevation dryland areas. How then can we explain its prevalence, maybe germination and establishment in abnormally wet years, or germination under nurse trees where water stress may be lower in dry ecosystems?
Yeah, that's a great observation. And that is actually like the main point. We cannot infer seedling drought tolerance from adult plant prevalence or distribution in the field. They're decoupled, and often the climate niches of seedlings is totally separate from the adult climate niche. And that's a really important point. And we're finding it, and people have found it in lots of other places too. So this is why relying on adult distribution to predict future distributions under climate change is so risky, because seedling recruitment can follow totally different rules. And yes, what can happen is that you could have a climate niche for seedlings that's outside of the climate niche of the adult stage. And that could be driven by just very infrequent episodic recruitment under unusually wet conditions. And that's actually not that uncommon of a life history strategy. The problem, I mean, we don't care about Christmas berry, right? But the problem for native plants that might be doing that is if we never get those really wet years, or we don't get them at the time that there's seeds in the soil, then we may not get that rare episodic recruitment event that the population requires for stable population growth. So that's really the concern, and why characterizing the climate niche of seedlings is really important. And I would also add the caveat that my species comparison is under those controlled conditions in the greenhouse. So that's potential tolerance, that's the intrinsic capacity of these plants. But the realized tolerance is going to be dependent on lots of other things. And that I can't speak to, like maybe Christmas berry has some mycorrhizal association that initiates really fast and enhances water availability, whereas other species it's slower. I mean, I don't know. Nurse logs, that's another good idea.
So this will be the last question. So thanks so much for the interesting talk. I'm thinking maybe measuring soil moisture in the field can better describe and compare to the greenhouse approach. I'm also curious if those plants survived from the drought increase. I'm also curious if those plants that survived from the drought increased their resistance to drought later, comparing to those that grew under normal conditions because of stronger.
Yes, you're totally right that measuring soil water availability out at our field site would have been ideal. Yes, I wish we had been able to do that. There's no doubt that would have given us more precision and would have been really helpful. We were we predicted that the initial drought would prime the plants, sort of pre-acclimate them to have better performance under the terminal drought, and we did not observe that. So we've tried this in a couple different experiments where we do repeated droughts to see if the plants sort of harden or become acclimated, and we have no evidence that they do for ohia. That's the species we focused on. So it does seem like ohia is just super tiny seedlings too, right? So they aren't adjusting in a way that then allows them better recovery from subsequent droughts. We see a sort of additive effect of multiple droughts where they just keep getting hit really hard and having high mortality. Ohia has actually some of the highest mortality under drought in our experiments across all the species. So it's interesting, the ones that survive, we've been able to characterize their physiological strategies, and they survive for a long time, but most of them die. And that's not, I only showed you the biomass growth data, not the survival data. So that's another piece of this that would be helpful to consider. Thanks everybody for so many good questions.
Let's thank our speaker one more time.